Good day, my dear postgraduate students. Welcome to R.A.T.S for PGs. We are racing against time. And this class is on gene for silencing. Again, a wonderful pair of genes which we would like to buy at an extra cost. Our class is on gene silencing as mentioned over here. Nice. A meaningful silence is always better than a meaningless word. Now, do you believe in God? I thought I would define God as such. I don't know much about it. But then God has said, whenever somebody does something wrong, I shall be there to correct them or to silence them. So these are the quotes of God, rather my own. So is this gene silencing? Maybe something like that. Before we plunge into the topic per se, a small recapitulation of it. You find that there is a cell and then it has got a nucleus which has got this clumps of chromosomes. And the chromosomes themselves have got this unwinding or entangled bundles of this nucleotides in the form of DNA, adenine, cytosine, thymine, guanine, etc. Basically, the functions of the genes shall be to have the components of the genetic material, the units of inheritance, to control the morphology of a phenotype, and to carry any hereditary information. Also, there can be reshuffling of the genes at sexual production to produce a kind of variation. This is somewhat, I thought, will be in the background before we go further into the topic. And this also, this particular diagram can be more useful for the molecular basis of thalassemia and so on. But basically you find that there is a promoter and then there is a transcription site, there is an intron, exon, and the transcription site again. 3-5-, etc. You people would have known. And after some time you find that all the unwanted ones are being removed by a process called as splicing. The intron sequences are being removed during this splicing. Finally, you get only the complete and perfect finished products of the exons. And this can undergo a bit of translation, a modification. Finally, you get the DNA protein that is completed. So these are the stages of a gene function, thanks to ResearchGate. Now, what is this gene silence? It can be considered to be a negative back in a very colloquial form. But then the definition is interruption or suppression of expression of a gene at the transcriptional or translational level. So there are two pictures you have seen. So when they are getting suppressed, you can call it as gene silence. This can occur at transcription or translation. And this is a brief history on this. 1980s, antisense oligodeoxynucleotides were found. 1990s, ribozymes. And 2000s, RNA interference. That is why the small I. So, gene silencing. Is it the same as gene knockdown or regulation? We shall see the difference. Now, there are two things over here. It looks more like a mathematical class. Gene analysis, two different approaches. The first approach is there is a normal gene leading to a normal function. In this case, there is a defective gene leading to an aberrant function. And this is being discovered. And finally, you find that the defective gene is being disrupted. So this is one form of, say, a gene therapy or kind of correction. In another one, you find that the aberrant function is first identified and the gene for it is identified further, and they are again made functionless. This is another one. There are two analyses which you people can, and this is an age-old picture of an optical illusion. What is the difference between the two? Which of these is young and which is old? And are you seeing two in the same? This is supposed to be a very wonderful young and the old woman optical illusion, and there's a history behind it way back in 19th century, which I found in the clinical medicine book of MacLeod. Now this again, we need not know much about it except that there is a nuclear membrane or an envelope over here within which there is a DNA. It undergoes a transcription, there is a processing, and there is an export of the nuclear material. This undergoes a maturation. Sometimes you find that there can be a dicer which helps in the maturation. And there can be the risk which can affect the larger messenger RNAs. This is a normal one that is happening. But at any point of time, you find there can be defects which should be silenced. Gene silencing and knockdown, are they the same? What is the difference between them? Gene silencing, the efficacy is reduced due to reduced expression of any particular product. Whereas gene knockdown means there is a complete erosion of it from the genome. Therefore, there is no expression at all. So you can say that gene silencing is somewhat partial, whereas gene knockdown can be complete. Distance doesn't separate people. Silence does. After all, this entire class is about gene silencing. And this is the history of the gene silencing. It has been obtained from the ResearchGate. I would like you people to kindly go through, look at the various things that have been contributed by various people. And in fact, there are a couple of Nobel Prize winners in this also. What are the types of silencing? There are three types which you people must remember. The first is transcriptional, and it has got certain subheadings such as genomic, paramutation, transposon, transgenic, position effect, RNA direct. So these things we will by heart. In all these cases, the promoters are silenced. Silence isn't empty. It is actually full of answers. Maybe that is why you people are silent at the exam. Now, I am just expanding this transcriptional silencing. Genomic imprinting means you find small proportion of the gene expression only from one parent is there. Example, insulin-like growth factor. It comes only from one parent. Paramutation, interaction between two alleles in one locus. Alleles means the adjoining regions. So there is an interaction. Transposon silencing, histone modification. Transgene silencing by the DNA gene or viral vector. You find that there is a silencing. Position effect, change in location. Translocation. This can lead to even a change in the color of the eye. RNA-directed DNA methylation. Small interfering RNAs. That is why it is called small s, small I. Small interfering RNAs are there. So this is one. If possible, you people, but at least the names of them we people should remember. This is a normal gene protein. There is a gene and there is a DNA, then there are the various nucleoproteins, there is a messenger RNA. Ultimately, there is a translation and a protein product is there, which is normal. In a case of gene silencing, there is transcription again, and you find that molecules that censor certain products of the RNA are being released. As a result of which, you find that it prevents translation, and you will not be getting a complete product of that. So this can be used for therapeutic purposes. And Comparison of the gene silencing strategies. Mostly it is drugs, and these can be the various ones which can be used. And what is the mechanism? It can bind to the protein, or it can hybridize messenger RNA, catalyze the messenger RNA, or the target messenger RNA. So these are the various mechanisms, and ultimately there can be either a degradation or an inhibition of the nucleoproteins. So this is one thing that we can remember. At least a couple of them. It will not be as difficult as it appears. Next one is post-transcriptional. In this case, there is an RNA interference. RNA molecules inhibit the gene expression. RNA silencing regulated by non-coding RNAs. Nonsense mediated reduces the errors. Most of these things are all new terminologies to me also. But only thing I know is in the case of RNA polymerase 3, I find that the transcript is folded upon itself. A hairpin bend is being created. Sometimes, since it is bending in the form of a finger, it is called a zinc finger. Meiotic is another one. A synapses, and this is the one. Sex chromosome remodeling is there, and finally there is a chromosome silencing that can occur. And this can be because of a synapsed chromosome pair. There is a DNA damage that is occurring, and it is being mediated by this particular substance. This is called as meiotic, the third one. So three types you people will have to remember. The therapeutic advantages are for combating cancer, for infectious diseases, neurodegenerative, as in this particular case. It is called as a Huntington disease. And I find that there is something abnormal with the DNA over here, blocking the expression of unwanted genes, and cost effective. It is a powerful tool for analyzing unknown genes in sequenced genomes. After all the genes have not been analyzed, and you are not expressing all the genetic properties within you. And this is a case of Huntington's disease, a neurological disease, and thanks to Child Neurology Foundation. Applications. It can be used in the treatment of AIDS, viral replication and culture. The infection can be blocked. Okay, and the human genes will explain the CD4. Also, you find it can be used for biotechnology and epigenomic analysis or for research purposes. In cancer, see what is happening over here. So you are giving a cancer treatment. There is a conventional treatment. This is a malignancy, conventional treatment. So the cancer cell, it gets affected, but the tumor can relapse. It again comes back. Whereas in this case, it is quoted that it is more efficient. There need not be a relapse, at least hypothetically. So it involved in the cell division. And also, as I mentioned earlier, it can be used for the treatment of any macular degeneration. Over here, you find that the injections can be subretinal or intravitreal. Treatment of macular degeneration, and the virus that is used is the respiratory syncytial virus. Surely, silence can sometimes be the most eloquent reply. Thank you.